about doing something like moving our hands and arms, and that electrical impulse will travel through our body, it will cause muscle contractions, and of course there has to be energy present for all of that to occur. What's so interesting is that there was work done by a scientist in California by the name of Joey Jones, and he was stimulating an acupuncture point on the big toe. And in acupuncture, this is known as a point that improves vision. So he had a hypothesis about this, and he found that when you would stimulate this point on the toe with light, it would, using a functional MRI, you could measure that this would light up the visual cortex. Now, how people thousands of years ago, how they knew that, that's something else entirely. But what he found was that the light signal through the meridian, and the acupuncture meridian is a collagen string that has minerals attached to it, that this would travel about 200 times faster than the electrical signaling through the nervous system. So we need to understand that the way that we were created was that light is going to initiate all the biochemical changes in the body. And the common example is we go out in the sun and light will cause our body to make vitamin D or whatever else. So the Lifewave patches contain a blend of natural materials. They're stereoisomers of amino acids and sugars. And they're processed in a very specific way so that when they're activated by body heat, they will stimulate the skin with very low levels of light. With X39, we want to do two things. The first thing is we want to upregulate mitochondrial function. So when the patch stimulates the skin with light, it will increase an enzyme in the cell called cytochrome C oxidase. This will in turn upregulate fat burning in the cell, and there's a signal that turns on production of GHKCU copper peptide. And of course, this in turn provides the body with all of these health and wellness benefits. So the patch is activated by body heat, stimulates the skin with light, and increases the body's levels of copper peptide.